resistant to exposure to the virus, and so the idea is to maybe try to scale this up so that we can genetically alter these, so people can genetically alter these pigs to be resistant to the virus and save farmers a lot of money and save the pigs in the farms. And also having to do with pigs resistant to exposure to the virus, and so the idea is to maybe scale this up so that we can genetically alter these, so people can do it for humans, because there's not really sufficient organ donations for humans that need organs, and pigs have certain qualities that make them a potential good donor for humans, but they would be rejected, and so there's an idea of modifying pigs so that their organs would be more acceptable to humans in organ donations. And so that's something that we'll also talk about later on when we talk more about other types of transgenic animals. Another thing that CRISPR can be useful for, in addition to like actually modifying animals or making drugs, is making animal models of human disease. So CRISPR makes it feasible to work with all sorts of animals, even large animals like monkeys, rather than only relying on smaller animals, which many times we rely on these smaller animals. And this how, studying larger animals is more difficult because of breeding and everything, but using the CRISPR technology makes it more feasible. And so making animal models of human disease is a really good way to study disease and try out different types of treatments. One ongoing study involves disrupting a gene for SHANK3 in monkeys. This gene has been implicated in some forms of autism, and so while this is a difficult disease to study in non-humans, monkeys that have disrupted SHANK3 gene, in which it's disrupted by using CRISPR, have some signs of autism to the extent that it can be studied in other types of animals, and the plans for this study are not just to delete the gene, but to delete different parts of the gene and to try to find out which, if any, part of this gene is particularly important, might be particularly important in some forms of autism. And then HIV is another area where CRISPR becomes very important. So this is the HIV virus. This is the HIV virus, and here it's invading a cell, or it's attaching to a cell. And so it binds to CD4+, and it binds to this receptor called CCR5, which is CCR5, which is important right now. So people have tried to understand how maybe one could use CRISPR to modify cells so that they are resistant to the HIV virus. And this has actually been tested to some extent. So scientists used CRISPR to induce a mutation in this CCR5 gene. So remember, that's the gene for the receptor for HIV. And they actually modified it in human embryos, which is quite controversial. But they wanted to find out whether they could mutate it, and they wanted to see whether it could actually, CRISPR could work in embryos. And in about 4 out of 26 embryos, the mutation was successful. So it's not a high rate, but they were able to be successful. And in the study, the embryos were from IVF clinics, in vitro fertilization clinics, where there are sort of stores of embryos sometimes, and these were not viable because of having an extra set of chromosomes, so that made this whole process a little bit more ethical, because they were not really viable embryos. But they found, the purpose of the study was to see whether you could, in fact, delete this gene in embryos, and then, with future plans to possibly modify embryos altogether. And in fact, in 2018 and 2019, 3 CRISPR edited babies were in fact made. I believe they were made in China, and so they found that the idea was because to make these babies resistant to HIV, I think it's because one parent carried HIV, but after making this had been outlawed. I think it was outlawed before. So now there's a worldwide moratorium on using CRISPR editing on any viable embryos, while the scientists deal with and think about the ethical implications. But you can see the implications are vast for the different types of things that people could potentially do with CRISPR. But in this particular study, the idea was to generate, in this case, a baby or person who would be resistant to HIV infection. Yes? This receptor, that's a good question. He asked what's the significance for this receptor. As far as I know, this receptor doesn't have any functions that life depends on. So it's a receptor for the virus, and one can survive without it. Wait. Okay, so first question about CCR5 gene. Right, so mutating, inactivating the CCR5 gene that would potentially make the cells resistant to HIV because this is the receptor for HIV. Okay, so another thing I wanted to talk about in relation to HIV is something called gene drives. And there's an idea to use gene drives to alter the wild mosquito population because mosquitoes are a very important spreader of human diseases such as malaria. And so malaria is a very devastating disease and actually quite common. It's caused by a microorganism called Plasmodium. This is a parasite that's present in the saliva of some mosquitoes. Plasmodium can then enter the body of an individual after they are bitten by the mosquito and cause malaria, which can lead to death. It's very severe in some certain places. The plasmodia can then spread again to other individuals. So this individual gets bitten and that can be spread to another individual. And so there's an interest in making genetic modifications that would either make mosquitoes resistant to plasmodia or maybe make the mosquitoes infertile altogether so that they can't replicate. And so it's very difficult to make a genetic mutation in a wild population compared to in a controlled population in the lab. And a promising way to do this and to spread a mutation in a wild population is through CRISPR. And so this is a quick video that explains why it's difficult to make genetic modifications in a wild population, and then we'll talk about how CRISPR can help with this. Okay.
Plants and animals, but have not been able to do this to wild populations. Here we use mosquitoes as an example to explain why most genome alterations designed by humans don't persist in nature, and how a recently proposed technology could change that. The transgenic mosquito, tinted blue, has an altered gene inserted into one of its chromosomes. When it mates with the wild-type mosquito, each parent contributes one copy of each chromosome to their offspring. Thus, only 50% of offspring will carry the altered gene, while the other half will inherit the wild-type version from both parents. Even if the altered gene doesn't reduce the likelihood of each mosquito surviving and reproducing, it may persist at a low frequency in the ocean of wild mosquitoes, or it might go extinct after several generations, especially unlucky inheritance. This process is what keeps us from altering wild mosquitoes to prevent them from transmitting diseases such as malaria or dengue. A team led by Kevin Esfeld at the Wyss Institute, the Harvard Medical School, and the Harvard School of Public Health has now outlined a way to build gene drives that can improve the odds that almost any altered gene will be inherited, potentially allowing them to spread through even wild populations. The proposal relies on the CRISPR-Cas9 system, a new genome editing technology co-developed by the same researchers at the Wyss. Gene drive mosquitoes carry both the altered gene, the genes for the Cas9 enzyme, and several guide RNAs that tell it where to cut. When passed to offspring, the guide RNAs direct Cas9 to cut the wild-type version of the gene inherited from the wild-type parent. The cell then copies both the altered gene and the drive when it repairs the damage. Because the mosquito now has two identical copies, one on each chromosome, all of its offspring will inherit the alteration and the gene drive. This same process will be repeated in subsequent generations, causing the alteration and the gene drive to spread through the population.
Okay, can you still hear me? Let's see. Right. Okay, so gene drives can be used for altering wild-type populations, where you can see from the last video that would be difficult to do with other more conventional methods. And basically a summary, this is showing a mouse, even though we're really talking about mosquitoes, but this drawing is showing would work the same way in a mouse. So one allele is going to have the whole CRISPR apparatus inserted into it by genetic modification, and then it's going, the whole CRISPR system is going to recognize a specific gene or gene segment that's found on the other allele on the other chromosome, right? So if you have the whole CRISPR system here, and then here's the wild-type allele that you want to edit, then within the egg cell of the mouse, or whatever cell of the mouse, this CRISPR system is going to cut out, it's going to do genetic, it's going to already have the genetic modification you want because you modified it, and then it's going to modify the same gene on the other allele, on the corresponding allele, right? Because every gene has two alleles, and now both alleles. So the idea is that by using this gene drives, you can make sure that the egg cell is going to have a homozygous mutation rather than a heterozygous mutation. And then the idea is that you can now mate, so you can make a mutant animal with a wild-type animal, and the progeny is most likely, if it works right, going to have this mutation on both alleles. Whereas normally, when we do genetic modifications, like with some of the techniques that we'll talk about next, you would just only have mutation on one allele. So the bottom line is that and so then you keep mating and mating and mating until the wild-type population has a high percentage of these homozygous mutant alleles. And so the main take home about the gene drives is that the gene drives are a way that you can get a homozygous mutation, whereas normally we would only have a heterozygous mutation if we mate one animal with another. If we mate a mutant animal, the wild type animal, its progeny are going to be heterozygous normally. But when we use these gene drives, we can increase the amount of homozygous mutants that you get in the progeny, right? And that's because the mutation on one allele will be repeated on the neighboring allele. Okay, and if it's not that clear, just watch the video one more time. You have a link to it and look at the image. It will, I think, will start to make more sense. And come see me if you're confused about it. But the idea is that one could potentially, mutate the whole genetic population of malaria-containing mosquitoes, either so that they have an alteration that makes them resistant to the plasmodium, or that they become, or something that makes them become infertile so that they can't pass on anymore. So that's another idea of something that people are interested in using gene drives for in order to actually manipulate wild-type populations. And then finally, we've talked a lot about using gene drives for drug development and for actually modifying animals for various purposes, but a very important use for CRISPR is just in basic research. So one of the most important uses for CRISPR is in basic research. So when people do research in the lab, they often want to study what certain genes do, what is their normal function in an animal, and so by using CRISPR, people can turn certain genes on and off and try to study their functions, right? One might be studying a gene that's important in animal development, or a gene that's important in the brain, or a gene that's important in some types of cancer. And by using CRISPR, one can turn the genes on and off, or mutate them, or mutate specific parts of them, and study these genes in more detail in an intact animal. So this is a summary of some of the really important and powerful things that one can do with CRISPR. And now we're gonna skip ahead. Everything after this is optional if you're interested, but not part of the exam. And now, in this lecture. And now we'll skip ahead to the next one that should be labeled lecture 26. Okay, everyone have this? Okay, so the next thing we'll talk about are just transgene. Let me turn this down because it's I think if I turn that down. Yeah, just kind of echoing. I think it'll. Okay. All right. So the next thing we talk about is more about transgenic animals. So CRISPR is one of the newest ways to make transgenic animals, but I haven't really discussed some of the other ways that people make transgenic animals and have been doing for years and still do, even though we have this CRISPR technology as well. So let's talk about some other methods that people use for transgenic animals generation, and also talk about why. So we'll talk about, and we'll go as far as we can today. We'll talk about farming. Farming with a PH has to do with making transgenic animals that can potentially produce drugs that can be used for humans. And then we'll talk about transgenic animals that can be used for xenotransplantation, like I alluded to a little bit already, about using animals as organ donors for humans. And then we'll talk about transgenic mice and knockout mice that are used largely in research and for making animal models of disease. Okay, so let's talk about farming. So farming has to do with using large animals like goats and sheep, and sometimes smaller like rabbits, but basically larger than a mouse, and using them as models for human disease, but also using them to produce drugs that can be used to treat human disease. So farming specifically has to do with generating and using transgenic animals to produce large amounts of drugs that can be used to treat diseases in people. And often the drugs are produced in the mammary gland, and in this way they can be expressed in the milk. So here's a few examples. So one example, and this is the first one that was not as successful as some of the others, but I just want to show you the basic method. So this is AAT, antitrypsin, and this is a drug, this study is to generate a drug that treats AAT deficiency, which is a disorder that can lead to damage to the lungs. And patients that have this disease need to replenish the AAT that they get, and it's difficult to get a large enough amount. So the way this system works is you make a transgene. So this is the AAT, this is a sort of very simplified picture of the gene. This is the AAT gene, the coding sequence of the gene, and it's fused to a beta-lactoglobulin promoter. So it's artificially fused to that promoter. It's not a normal promoter, but people generate a transgene where you take the gene and fuse it to an exogenous promoter. And the reason for the beta-lactoglobulin promoter is because beta-lactoglobulin promoter will regulate where the gene is expressed, and particularly will control that the gene is expressed in the mammary gland. So in this example, they use the pro-nuclear injection method. So this is a zygote right here. It's a zygote which has two nuclei, one from the egg, one from the sperm, that are soon going to merge. At this stage, this is where they found the best stage for nuclear injection. So they inject the transgene into one of these pro-nuclei in the zygote, and then very shortly afterwards, the nuclei will fuse together. And now you have an egg. So this is a newly fertilized egg. It's the only cell in the embryo at this point. You inject it with this transgene, and then the egg is implanted into a female animal that's been treated with hormones so that they can take up the egg and produce an offspring, and then they have an offspring. The offspring is going to, if everything worked as it's supposed to, some of the offspring at least is going to carry this transgene. The transgene, because it has the beta-lactoglobulin promoter, it will express the gene, meaning it will transcription and translation will occur, and it will occur specifically in the mammary gland because of the beta-lactoglobulin promoter. Because it's produced in the mammary gland, it will be expressed in the milk. And so the idea is that it's relatively straightforward to produce, get milk from a goat, get the milk, and then you can and then isolate the protein from the milk. And people have done some clinical trials to take this AAT from the milk and insert them and give it to patients who have difficulty getting enough of this protein in other sources. And in this particular case, there were abnormalities in the glycosylation of the transgenic protein. So the protein was there, but it wasn't exactly modified correctly, and it got removed from the circulation too quickly. So this is not this particular project was not that successful in treating patients, but it gave them the technique basically worked in terms of getting large amounts of protein produced in the mammary gland, and so people kept using this type of technique to make other types of drugs and proteins. Okay, so here's the next question. Specifically is the beta-lactoglobulin promoter. So the answer is the beta-lactoglobulin promoter. That's it. That's what's driving its expression. So presumably, one could take any gene and fuse it to the beta-lactoglobulin promoter, and it would most likely be expressed in the mammary gland because that's the job. The promoter controls all these different things like where and when a gene is expressed, and this particular promoter drives expression into the mammary gland. Yes.
Yeah, I have like around 10 ADP. That would leave us pretty good.
He's asking why we can't include the AAT gene. Without the AAT gene, it wouldn't be produced at all. But I guess the question is based on not why it's produced, but why it's produced in the mammary gland, as opposed to any place else. Right, without the AAT, it wouldn't be produced at all, but the point of the question is why is it specifically in the mammary gland, as opposed to, the skin or the brain or someplace else. Okay, yeah. Well, if it's on the test, because you reminded me that it's a little bit confusing, I'll make sure to clarify it. Okay. Okay, so next, I think if I was on the test, I might say, why is it specifically expressed in the mammary gland? So that would, focus not why it's expressed, but why in the mammary gland. Okay. Okay, so next drug and next transgenic animal involved in farming is something called antithrombin. It's also called Atrin or AT3, and this was produced in transgenic goats. So antithrombin or AT3 is a regulator of blood clotting. Some patients have an AT3 deficiency, and these patients have an abnormality in blood clotting, and this can be a big problem in patients, especially under circumstances where they lose blood, such as in surgery or even childbirth, and there are situations where they need to have extra AT3 AT3 to make up for the deficiency they had. And there's different ways that these drugs can be produced, of course, but I'm really focusing on the transgenic animal method. So AT3 has been produced in the mammary glands of transgenic goats by a similar mechanism that I showed you before. So, and this drug was actually FDA approved quite a long time ago by now, and it's used for treatment of antithrombin deficiency in patients undergoing surgery or childbirth. And it's actually the world's first transgenic animal drug, and it's already been used quite successfully in patients. AT3 is made from transgenic goats. It is significantly more efficient than some of the other ways that patients have been getting AT3, such as by blood transfusions. Quite a large amount of medicine can be generated from these transgenic goats, and there's actually a herd of these goats kept on a farm, sort of a lab farm in Massachusetts, where they can obtain as much of this AT3 as they are able to. So AT3, or Atrin, is an FDA-approved drug that's made in transgenic goats. Another drug, Ruconest, is another transgenic animal drug. It is used to treat a disease called hereditary angioedema, or HAE. It was approved also it's also FDA-approved drug. The disease is called HAE for short. This is an inherited disease that associated with episodes in which fluid accumulates outside of blood vessels, causing swelling and sometimes other severe side effects. It's caused by low levels of a C1 esterase inhibitor. So the normal role of the C1 esterase inhibitor is to control the levels of C1 protein, which is a part of the complement system, and we won't go into details about how this whole thing works, only to discuss really how people have tried to replace it. So the lack of this inhibitor leads to unchecked C1 levels, which in turn leads to unchecked levels of bradykinin, which is a vasodilator. Replenishing the C1 inhibitor can be used to treat patients with this disorder. And one way that patients can get more of this inhibitor is by blood donations, but this can lead to other side effects and problems and increase of risk of other diseases that people worry about when they get blood transfusions. A recombinant production of C1 inhibitor in bacteria has also been tried. So you can use bacteria to produce human protein sometimes, but this has been insufficient. The E. coli bacteria protein doesn't get glycosylated, which is an important modification of this protein, and so it doesn't work well. So more recently, the C1 inhibitor has been produced by generating it in transgenic rabbits. So before we were talking about goats, now we're talking about transgenic rabbits, but it's isolated from the milk of the rabbits, and this form of the enzyme is glycosylated, as it should be, and it's effective in patients, and it can be, patients have used this for quite a few years now, and they administer it by self-injection. Okay, next question. Right, so AT3 is one of the drugs that was made in transgenic goats and produced in the milk of the transgenic goats. Now, a lot of times these drugs are produced in the milk. There have been some situations where the drugs can be produced in the eggs of animals that lay eggs and other things, but a lot of times they're generated in the milk. Okay. So that's farming. So farming is the idea of using large animals to generate large amounts of proteins that can be used for human patients that need to be supplemented with those proteins. And it can be an advantage over some other methods such as blood transfusions or making the protein in some other way. So in some cases, using transgenic animals in the farming method has been advantageous. And often it involves goats, sheep, it can also involve other animals and even chickens as well. Okay, so the next part, let's talk about xenotransplantation. So xenotransplantation has to do with organ donations. So organ donation, the need for organs is a big problem. There's more of a need for donated organs than there are humans that are signed up to donate them. And so there's a constant need to find other ways to get organs, to get donated organs other than from other people that are not always available, which would be the first choice, but it's not always possible. So let's talk a little bit about some history of organ transplantation. So in the early 1900s, doctors tried to replace failing human organs with those of pigs, goats, lambs, monkeys, and all of them failed. In the 1940s, by the 1940s, the doctors learned that this failure was due to rejection by the immune system. Then in the 1950s, the first human-to-human transplant was performed, so that was also difficult to get working, but this consisted of twins. So it consisted of a twin transplant from one identical twin to the other, and therefore was successful because of the genetic identity. And they realized that, without being twins, it was difficult to get an organ donation because the organs would be rejected. In the 60s, immunosuppressive drugs were identified, and this made a big difference because using these drugs led to better outcomes, and it helped the patient, it helped suppress the immune system so that organs would not be rejected as readily. But nevertheless, there was still a problem of not being a sufficient number of human organ donors available, so people still were interested in trying to find a way to use animals as organ donors. So even as early as the 1960s, doctors tried using animals to transplant organs. They transplanted kidneys from baboons into 6 human recipients. The patients survived 19 to 98 days, but the success rate was low. In just a couple of years ago, a patient received a heart transplant from a genetically modified pig that was generated from a company called Revivicor. This patient survived 2 months with the transplanted heart, and the success rate from animals to human transplantation has remained low, but the researchers in this area are still hopeful that there are ways that one could modify these animal organs to be used in people. So when talking about organ transplants from animals to humans, pigs are often the animal of choice. So porcine xenografts would be organ donations from pigs. Domestic pigs are considered one of the best alternatives for organ transplants next to humans, which obviously would be better. So why pigs? Why are organ transplants from pigs so promising? So it has to do with anatomical, physiological, and biochemical compatibility. Compared to other animals, there's more compatibility with pigs. They're also more genetically modified, more readily genetically modified than primates, which would also be a good source. And then there's breeding, rearing, cost, and ethical considerations when you think of breeding pigs for organ donation versus breeding maybe monkeys or other primates for organ donation. There are a lot of things that make breeding pigs a more viable option. So in order to get successful xenotransplantation, what needs to happen? You need to overcome immunological hurdles. You need to find a way to prevent the transmission of pathogens. And then you need to find a way to ensure compatibility with the human anatomy and the human physiology and basically in the end, prevent any type of rejection. So, 1 just taking a normal pig organ and putting it into a human is not going to be very successful. So this company is one of the major companies in studying this, with Revivicor, and they have focused on using pigs for regenerative medicine, particularly for organ donation for humans. And so a major way that they are studying this and trying to make this work is by nuclear transfer technique. And we'll talk about that technique even more next semester when we talk about stem cells and some of the ways people study stem cells. But for now, the idea is they take cells from the pig and they genetically modify the cells. Now they can do it with CRISPR. In the past, they used other types of methods, and they genetically modify these cells in ways that they think will make the cells more compatible with humans, right? So they genetically modify the cells, and then they take an egg. They take an enucleated egg, so an egg without its nucleus, and they take the nucleus from this modified cell and put it into the enucleated egg. So now you have a new egg that is genetically modified. And they take this egg, they put it back in the pig, and hopefully the progeny of the pig will now contain this genetic modification, and one could get organs from these pigs, tissues, cells, both for studying and hopefully for human transplantation. And again, for this basic technique, this is basically all you need to understand about it now. Next semester, when we talk about stem cells and more about nuclear transplant, we'll go into this type of method in more detail. But the idea is to use this method to genetically modify the pigs so that they can have organs that will maybe be more compatible with humans. So what are some of the different modifications that they made? So this company has made all sorts of different modifications. I'm just going to talk about one of the modifications. So they looked at to see what are some of the reasons that pig organs are being rejected by humans, and tried to alter those things in the pig. So one of them was that pigs have a sugar called galactose alpha-1,3 galactose, or alpha-gal for short, or sometimes I just call it gal for short. Pigs have that on the surface of their organs, whereas humans do not. So humans produce antibodies against pig gal, and so as soon as a human will get an organ from a pig that's not modified in any way, it will immediately start to reject the organ because of the antibodies that regulate gal. So one of the things that this company tried to do was to generate a pig that does not, to do genetic modification so that they can generate a pig that does not have gal on the surface of its organs. So they generated transgenic pigs in which alpha-gal can no longer be added to the surface of the organ. And so these would be the normal organs. You can see they have the gal on top. Here would be the organs from the transgenic pigs in which they modified a certain gene that's responsible for adding gal onto the organ. So the idea is that they modified these pigs, they made them so that they don't have gal on their surface, and then they tried to use primates as a model. So they took organs from these pigs and put them into primates before they tried any of this on humans, of course, and primates survived initially 180 days with the porcine organs, which is better than before, but still obviously not good enough. So step by step, they're trying to find different things that they can modify to make the genetically altered pigs good donors for humans. And there have been various levels of success over the year. So transgenic pigs were generated with a series of modifications, like the modification I showed you, and then some other modifications that this group has determined would help make the organs more acceptable to humans. And then they tested these first in primates, and more recently they've also tested them in humans. So short-term rejections have been improved, so that, for the short-term, patients and primates can survive, can keep the organs much longer than they did before, but over the long term, there's still eventually rejection, which remains a problem that they're still working on. In 2021, a genetically modified kidney from this same company was transplanted into a patient. This was a patient that was already basically brain dead, but on life support, and the family gave them permission to transplant their kidney just basically as a way to test it, and they found that the kidney lasted 2 months, and at that point they removed it so they could have a burial for the patient, so I don't know if it would have lasted longer, but that kidney was able to last 2 months in that patient. Later on, just a year ago, a patient received a genetically modified pig heart, also from this same company, and normal heart function was seen for some time. The patient survived 2 months. The patient did eventually die after 2 months. It's not clear how the patient, what was the cause of death, was due to the heart or anything else. Very recently, just a few months ago, a second heart transplant patient was generated with a modified pig heart. I think that this patient is still doing okay, but it was very recently, just a couple months ago. So that's where the status is with this particular project. So scientists are continually working on finding the best genetic mutations and trying to reduce long-term rejections, which are still, I think, the most serious problem. Okay, next question. Right, so the nucleus from a genetically modified pig cell is inserted into an enucleated pig egg, right? So basically you're making a new egg that has the genetic modification. Okay? Okay, so we talked about farming, using large animals to produce drugs. We talked about a few examples of xenotransplantation, genetically modifying pigs for organ donations, for the potential use for organ donations. Now I want to talk about transgenic mice and knockout mice. That's one of the most common types of transgenic animals used. People do them in the lab for all sorts of purposes, to study genes, studying mice is obviously much easier than studying large animals like goats and sheep and pigs. So studying transgenic animals in the lab is quite common as a way to study the functions of various different genes. So when we talk about transgenic animals in mice, there's two major things that we talk about, the transgenic mouse and the knockout mouse. So the transgenic mouse is a mouse in which a transgene is inserted into it, usually via the pronuclear injection method, like the method we talked about in goats, but in mice. And then there's a knockout mouse. So the knockout mouse is technically maybe not a transgenic mouse because you're removing a gene instead of adding a gene, but we usually discuss it together with transgenic animals too. So in a knockout mouse, you actually eliminate the function of a gene, and then you can either eliminate it in all the cells, or you can eliminate it in only some tissues. And this uses the genetic homologous recombination and the ES cell method, which we'll talk about. And that can be used for studying the function of a gene. What happens, you have a gene that you want to know what its role is. You remove it from the animal, and you can study what its function is. Both of these are good models for studying the functions of mammalian genes and also for modeling human diseases. So I'll talk about some examples of these and how they work. Before I do, one of the early transgenic mice was made in the 1980s, and since then the field has really grown, and many people make and study transgenic mice. This was a study in the 80s where people inserted the gene for human growth hormone into mice, and you can guess from this picture which one has the human growth hormone in it. So you can see that the human growth hormone made this mouse much this transgenic mouse much bigger than its control mouse that doesn't have human growth hormone. So this showed the researchers that implanting a gene from a foreign gene into an animal, the gene can still function and carry out some of its normal functions, in this case decreasing the growth of the animal. So how are transgenic and knockout mice made? So first let's talk about knockout mice. So the common method for making a knockout mouse, and some of these things will change with the advent of CRISPR and everything, but the common method of making a knockout mouse involves using embryonic stem cells, or ES cells. So embryonic stem cells, and again, next semester we'll have a whole much longer discussion about stem cells, but embryonic stem cells are the stem cells that come from a very early embryo that have the potential to become all the different cells in the embryo and eventually in the whole body. So these are embryonic stem cells from a mouse. So everything's from a mouse here, except for the foreign DNA that can be from anything. The foreign DNA is inserted into the embryonic stem cells. In the case of making a knockout mouse, the foreign DNA is usually a type of a DNA sequence that will actually cause the deletion of a gene, which we'll get into later. But you put in this DNA into the embryonic stem cells. So not all of the embryonic stem cells are going to take up the foreign DNA. So you select, there's various ways that people can select for the stem cells that took up the foreign DNA. And now you take a blastocyst, which is a very small ball of cells, which is like a very early embryo, like 3 to 4 day stage embryo. You inject into this blastocyst with a very fine needle and very carefully the embryonic stem cells that contain the foreign DNA. And then you take this blastocyst, you transplant it into a female mouse, and the female mouse has progeny. You take these progeny and you analyze them, you do genetic testing on them, and you see which ones have taken up the foreign DNA, and then you breed them with each other until you get a mouse in which all of its cells come from this, basically this cell here that has the foreign DNA, and then you can analyze this mouse. The other method, yes? The DNA, he's asking the source of the DNA for, yes, it's in a plasmid, and I think in a few minutes I'm going to show you a little bit more detail about what the DNA is and what's the significance, yeah. So we'll get there. The other way to make so usually this method is used for making a knockout mouse because the DNA, and we'll get to that in a minute, is in a form that will cause a knockout. Will cause a gene to be deleted. And the other method is a transgenic animal where usually you're trying to overproduce a gene. So here, we talked about this method already. You have this fertilized egg with the 2 pronuclei. You inject into one of the nuclei the desired DNA, the DNA you're interested in, and then you transplant that back into the mouse and analyze the progeny. So the pronuclear injection method is usually used to overproduce a gene, whereas the ES cell method is usually used to knock out a gene. So, and then the other thing you need to think about is random versus targeted insertion of a transgene. So if you're inserting a new gene, is it just going to go in, where is it going to go into the new genome of the new cell, of the host cell, right? Is it going to just go randomly any place, or is it going to be targeted specifically? And so there's different ways that you can control for this. So when you use the pronuclear injection method, you usually get random replacement of the transgene where the transgene just basically can go in anywhere. But if you use the ES cell method combined with homologous recombination techniques, this can lead to targeted integration of the transgene where the transgene goes into the exact place that you want it to go. This is often the method used to generate a knockout mouse. We'll get to in a minute. In this case, the gene is targeted to a specific place in the genome. So you might even be replacing a healthy gene with a mutated gene in order to study it, for example. The transgene will replace the endogenous gene, right? So the endogenous gene is the normal gene that's already there. The transgene is the gene you're putting in. This can be used to restore the function of a mutant gene, but it can also be used to knock out the function of a normal gene in order to generate a knockout mouse. Okay, so let's talk about the knockout mouse. Okay, so this is where where you asked what's the source of the DNA. So let's say, this is a certain gene that you want to knock out. It's sort of a simplified view. This is the gene, this is the upstream sequence, this is the downstream sequence. And so you're going to make a vector within a plasmid that contains the upstream sequence, the downstream sequence, but instead of the gene, you have some other sequence. In this case, it's a neomycin resistance gene, which is, so basically the idea is to replace this gene with the neomycin resistant gene. So this gene is gone, but you can tell if your cells have the gene that you want by growing them in neomycin and seeing if they're resistant. The outcome after homologous recombination takes place, so this all happens in the cell. Yeah. The cell already has this, you're inserting this vector into the cell. The desired outcome is that you have this new gene where this gene is replaced with neomycin, right? So the upstream sequences remain the same, the downstream sequences remain the same. Homologous recombination makes that happen, but the gene is replaced with the neomycin resistance gene. So basically this is a knockout mouse, because even though you've added a new gene, the main goal you accomplished was to replace was to get rid of a gene, to delete a gene that was already there. So you deleted this gene that's colored pink here. A lot of times you don't necessarily want to delete the whole gene, because sometimes it's more difficult to delete a very large sequence compared to a small sequence. So here's an example of a gene that contains 8 exons. This is an example that came from my lab actually a long time ago. Instead of deleting all of these exons, a vector was made that had the sequence upstream to exon 1 and some of the sequence downstream to exon 1, and the only thing that was replaced was exon 1. But because exon 1 contains the start codon and the ATG, if you replace exon 1 with something else completely, you're usually going to knock out the function of the entire gene, unless there's another start codon somewhere else. So just by replacing the first exon, you can delete the whole gene. You can delete the expression of the whole gene. And this method is just more straightforward because sometimes it's more complex to delete a whole big, large section of a gene compared to deleting a small part. But you may still have this, you generally still have the same outcome where you delete the production of this protein from this gene altogether because of losing the start codon. Okay, so what we just talked about, if we're going to make this vector, so let's say we made this vector, we got this outcome of this basically modified gene, modified inactive gene. This was all done in the embryonic stem cells, right? So we put this vector into the embryonic stem cells. This is the wild type locus that was already in the embryonic stem cells. Now you've made new embryonic stem cells that have this new modified DNA. You take those embryonic stem cells, so that all happened right here, right? You take the embryonic stem cells that have the change that you wanted, you can grow them on neomycin to make sure that they took up the transgene. Then, just like we showed before, you take those cells that have the modification and you put them into the blastocyst, and you put the blastocyst into the mice. Okay, and then you analyze the progeny. The progeny at first are going to be chimeras, where some of the cells took up the transgene and some of them didn't, but you breed them with each other until you get progeny that took up all of the transgenes, that took up all of the ES cells. The target, I think you're asking, so I'm not sure what you're asking, but the idea, here. So this is being put into embryonic stem cells, right? So this is happening in embryonic stem cells. The embryonic stem cells are put into the mice. The progeny mice, this can go into any of the cells, but you mentioned germ cells, and it's important that at least some of the germ cells picked up this modification. Sometimes you do the experiment and none of the germ cells picked up the modification, and then you have to start all over. And the way you are going to know if the germ cells picked up the modification is that you're going to breed these mice with another mice and see if the progeny also picked up the modification. So if the germ cells don't pick it up, because it's a chimera, like I said, if the germ cells don't pick it up, then you basically have to start all over. So your goal is that at least in some of the germ cells are going to pick up this modification that was in the ES cells. So that's an important question, right? So the germ cells are critical here. So when I talked about, just going back here, this vector here that I showed, this is a vector for making a knockout mouse, a conventional knockout mouse, where you're knocking out one exon, which is going to make the whole gene inactive. But sometimes, and once you analyze the progeny, so you go back here, you analyze the progeny are eventually going to have the gene inactivated in all of their cells, right? You're going to start off with an animal that might not have it. There might be a chimera that has just taken up some of the ES cells and some of its cells, but once you breed it, you get a progeny in which all of the cells have picked up this modification. But sometimes you don't want the knockout mouse to have the knockout in all of its tissues. You just want to see the knockout in some tissues. And for this, you use something called the Cre-LoxP system. So Cre recombinase and LoxP are part of the Cre-LoxP system. And this is a way you can make what's called a conditional knockout mouse, where the gene is knocked out in only certain tissues. Like maybe you're studying the liver, and the gene is knocked out only in the liver cells. Or maybe you're studying the brain, and you want to knock it out only in the brain. So a conditional knockout, instead of knocking out the gene in all the cells, you knock it out in certain cells. And this is the Cre-LoxP system, is the way people usually do this. The Cre recombinase is an enzyme. It's originally isolated from bacteriophage, and it contains DNA that's not found normally in animals. And then LoxP sequences are short sequences also found in bacteriophage, not normally found in animals. Cre recombinase will cause two LoxP sequences to recombine with each other, and the intervening sequences are eliminated. So like this, let's say this is your gene. So this is your gene of interest. So this is, again, sort of a more simplified view of the gene, upstream, downstream sequence. Now you're going to genetically modify your gene using the same types of methods that I just showed you before, but instead of knocking out the gene completely, you're just going to insert two pieces. So it's going to insert a LoxP sequence here and insert a LoxP sequence here using that same type of homologous recombination method. And then, if there's a situation where Cre recombinase is present, this LoxP and this LoxP will combine together and the intervening sequence will be excised. So the genetically modified mouse that is being made has two LoxP sequences on either side of the sequence that you want to conditionally delete. And basically you could also do this. This slide is basically exactly the same thing I just showed here. They're just knocking out one exon though. They're knocking out exon 2. So LoxP sequences here, LoxP sequences here on either side of one exon. Then you add the Cre recombinase, and I'll show you how you add the Cre recombinase later. But once the Cre recombinase is added, you lose exon 2, and now you have a modified gene. Okay, so Cre recombinase, the Cre recombinase LoxP system is for making a conditional knockout, where you knock out the sequence in between the two LoxP sequences, and I'll show you in a minute how that makes it conditional. So basically, the way it makes it conditional is by breeding. So you make one genetically modified mouse, which is shown here. Here's your target gene, and you use these homologous recombination methods to make it with to have this target gene have two LoxP sequences, one on either side of the target gene. And then you have successfully made this mouse, so it's not a knockout. The LoxP sequences usually are put in a way that it doesn't have any effect on the function of the gene. So this mouse is probably going to look pretty normal. And then you breed this mouse. You breed it with a mouse that's a transgenic mouse that has the Cre gene for Cre recombinase, fused to a promoter that's a cell type specific promoter, or you could call it a tissue specific promoter, that causes Cre to be produced only in certain cells, right? So this mouse is only going to produce Cre in certain cells. So for example, let's say that it only produces Cre in the liver. So you might you make this mouse with this mouse. Now you've got your progeny mouse that has both the Cre recombinase and the target gene that's fluxed. When you have a loxed P allele on either side, the loxed P sequence on either side, you call it a fluxed allele. So you have your fluxed allele and your Cre recombinase. So in this mouse, in the specialized cell type, so I gave the example of liver, here you're going to have Cre produced, so the Cre is going to cause a recombination event, and the gene is going to be knocked out. But in all other cells, there's no Cre, and so the gene will not be knocked out. So this is a tissue-specific deletion. In your specialized cells, the gene gets knocked out because it was found it was in the presence of Cre, and in all other cells, the gene is still normal. So that's a way you can make a conditional knockout where the gene is deleted in certain cells, but not in other cells. And it all depends on this mouse that you're breeding your mouse with. So this mouse has a tissue-specific promoter, and you're going to look, sometimes you can buy these mice, you can make the mice, but you want a mouse that specifically produces Cre in the tissue that you're interested in. Is that clear? Somewhat? Okay. Next question.
What happened? That's right. I have no clue. I'm not even opening the presentation.
Right, it's to make a tissue-specific knockout, so to make sure to delete the gene in a tissue-specific manner. So the Cre LOX-D system, the important thing about the system is that you can make a conditional knockout mouse. Okay, so I want to talk about a few examples of knockout and conditional knockout mice that people have generated in order to study the functions of certain genes. So here's an example of a knockout mouse study that was used to study learning and memory. This was done quite some time ago, but it's a good example of how people used a conditional knockout mouse and a transgenic mouse to study a specific function. So some very rudimentary biology of learning and memory. This is a presynaptic cell. This is a postsynaptic cell, and learning and memory involves, the transmission of information from this cell to this cell. Transmission information goes down the axon, and from the presynaptic cell to the postsynaptic cell, goes through the presynaptic space. And within the presynaptic and postsynaptic cells are specific receptors that are involved in transmitting the signal. So in this study, they studied one of these receptors, which is called the NMDAR, or the NMDA receptor. Okay, and this NMDA receptor is found in the postsynaptic cell, and it receives signals from the presynaptic cell. Okay, so all the details of how the signaling works and how the receptors work, we're not going into, but I wanted to focus on the fact that they're studying this NMDAR by using an NMDA receptor by using transgenic mice. So NMDA receptors are found at synapses, and they are thought to have important roles in learning and memory. So the NMDA, so this study I'm going to talk about has to do with using knockout and transgenic mice to study the NMDA receptor in some more detail and study what its function is. So this is the basic molecular structure of the NMDA receptor. It contains two major subunits, which are called NR1 and NR2. And the NR2 subunit can take on two forms, called NR2A or NR2B. And this is due to expression of specific genes, either the NR2A gene or the NR2B gene. And when the NR2 NR2A and NR2B gene are expressed has to do with the age of the mouse or individual. So in this study of the NMDA receptor, the scientists made a knockout mouse of the NMDA receptor, of part of the NMDA receptor, and then they also made an NMDA receptor transgenic mouse, which is basically the opposite. They overproduced part of the NMDA receptor. So we don't have that much time left, so we'll go into this in sort of, not a lot of detail, but just go over basically what they did. So the knockout and transgenic mouse studies were done to study the function of the NMDA receptor, or NMDAR. So this is the knockout mouse, where, and all these details, you don't have to go through all the details, except for what we discussed today. So this is the knockout mouse, so they use the embryonic stem cell method that we talked about already to make a conditional knockout of the NMDAR. And then this is a transgenic mouse where they made an extra copy, they inserted an extra copy of a part of the NMDAR. Okay, so in the first study, they made a knockout of part of the NMDA receptor. So they take the mice and then delete the NMDA receptor. What they found was this deletion was embryonic lethal. What does that mean? That means that if they delete this gene, that no mouse can survive past embryogenesis. So that means something in this gene is necessary for the whole mouse to survive in the first place. And that's one reason that people sometimes make a conditional knockout mouse. If you find that a deletion is embryonic lethal, you might need to make a conditional knockout mouse so that the mouse can even survive long enough to be studied. So because it was embryonic lethal, they made a conditional NMDA receptor knockout instead. So for this study, they used the NR1 subunit of the NMDA receptor, and they knocked it out in a specific region called the CA1 region of the hippocampus. They chose this region because this region appears to be important for learning and memory. Okay, so they took the gene, so rather than memorizing all these small details in this image, I'm just going to, outline the important things that they did, which is what you need to know. So they took the gene for the NR1 subunit of the NMDA receptor, and this was flanked by LOXP sequences. So they made that modification, so it's now a floxed allele like this. And then they attached in a different mouse the gene for CRE to a promoter that only operates in the CA1 region of the brain. So this is the second mouse. So mouse 1, mouse 2. And so they mated mouse 1 with mouse 2 together, and then the offspring had the floxed allele and the CRE. And in the hippocampus of this mouse, they will have the NR1 subunit deleted because of the CRE LOXP, and in all the rest of the mouse, the receptor is normal. Right, so this is a conditional deletion of the NR1 gene specifically in one region of the hippocampus. So what were the results? So these mice, they lost the capacity to change the strength of neural connections in the CA1 region of the brain. They exhibited abnormal spatial representation and had poor spatial memory. They could not as readily remember their way around the maze. And they showed impairment in several other non-spatial memory tasks. So these experiments supported the investigators' hypothesis that the NMDA receptors are important and crucial for memory. So in the second study, they made a transgenic mouse which overproduces one subunit of the receptor. So the NMDA receptor can be made of NR2A or NR2B. And what they found was that, so people have already found that the NR2B subunits are usually found in young individuals, whereas the NR2A subunits are usually found in older individuals. And basically what they found, I won't go through all the details because we are out of time, but basically what they found was that if they made a transgenic mouse that overproduced the subunit that's usually found in the younger individuals, that the mice could have an increased ability to learn. So there's something about this NMDA receptor to learn and have memory. So there's something about this NMDA receptor that's necessary for learning and memory, and that one specific subunit of the receptor is particularly can lead to increased learning and memory. And then we're going to skip all of these, but I just want to, let's go through one more question. Because it's embryonic leaf. That's the main reason. Okay, so we'll only be responsible for up to what I covered today, and good luck. Thanks everyone, and I'll make an announcement about what you will be arrested.